This conference will now be recorded.
Hello everyone, my name is Viv Marks. I am currently working on my Master's of Biology Education at Grand Canyon University, and today I want to talk to you about the structure and function of a single synapse. What exactly is happening when our body registers that we need to move? You place your hand on a hot stove and what? How does our body tell our brain to move away? In this presentation, we will look at how electrical and chemical signals communicate information through cells. This presentation is for general psychology or introductory bio or chem students meant for higher education. First, we will define important phrases that will reappear in the neural communication process and identify a neuron. Next, we will discuss action potential and how electrical signaling happens. Then we will apply the information from the previous slides to the structure of a neuron and in detail how a signal goes from electric to chemical. Finally, we will discuss the consequences of a chemical synapse. To begin, we must understand what a neuron is. A neuron is a nerve cell specialized to initiate, propagate, and transmit electrical signals, typically consisting of a cell body, dendrite, and an axon. In short, it's a specific type of cell. Our bodies have muscle, skin, bone, blood, and nerve cells. We are focusing on one particular type of cell. Unlike the typical round cell, neurons have multiple parts that host different functions. The axon and dendrites are the communicative parts of the neuron. The cell body hosts the organelles, including the nucleus, where DNA is stored. The focus of this presentation will be on the axon terminal and the dendrites. Other important vocabulary one should be familiar with is neurotransmitter and ion. A neurotransmitter is the chemical messenger released from the axon terminal of a neuron in response to an action potential that influences another neuron or an effector with which the neuron is anatomically linked. Whew. To summarize, a neurotransmitter is the chemical that relays a message from one neuron cell to another. An example of a neurotransmitter is dopamine, which is a self-stimulating neurotransmitter. Drugs that combat psychosis, like amphetamines, stimulate the release of dopamine from neurons. Ions are atoms that have gained or lost one or more of its electrons, so it is not electrically balanced. In essence, it's an element in its smallest form that holds a positive or negative charge. There are patterns on the columns of the periodic table that identify ionic charges, which are the rules for the formation of compounds. The charge of the ion is important to the membrane potential of the neuron cell. Cells have atoms inside and out that maintain homeostasis. When homeostasis is skewed, the body works to remain balanced. Membrane potential is a separation of charges across the membrane. A slight excess of negative charges lined up along the inside of the plasma membrane and separated from a slight excess of positive charges on the outside. A cell's membrane separates the insides from its environment. The membrane potential looks at the electrical charge brought about by ions inside and out of the cell. Membranes have pores or channels controlled by different factors that will allow water, ions, or other material to enter. Synapse and action potential are the final vocabulary words with impact. A synapse is the specialized junction between two neurons where an action potential in the presynaptic neuron influences the membrane potential of the postsynaptic neuron, typically by releasing a chemical messenger that diffuses across the small cleft between neurons. This specialized junction is the location where the axon terminal of the axon of one neuron meets the dendrite of its neighboring neuron. One neuron releases a neurotransmitter like dopamine towards the other neuron. Now, we've seen action potential a few times so far, but what is it? Action potential is a brief, rapid, large change in membrane potential that serves as a long-distance electrical signal in an excitable cell. We know that membrane potential is the positive and negative ions on either side of a cell's membrane. Action potential is the opening or closing of the pores of the membrane that alter chemical equilibrium. First, the cell allows sodium ions to enter the cell. They are positively charged and increases the membrane potential depicted here in phase 1. While sodium ions enter, potassium leaves. Potassium is also a positive ion. This repolarizes or brings the internal charges of the cell back to a resting potential, as seen here in phase 2. Action potential influences membrane potential from our hands up to our brains. When action potential increases in one cell, the sodium and potassium ions move through the body to create a domino effect of cell communication. How that happens is through the synapse of neuron cells. The action potential travels through the neuron until it reaches the synaptic knob, a bell-shaped ending to an axon terminal. Inside the knob are vesicles that contain the neurotransmitters. When the action potential reaches the axon terminal, it opens voltage-gated pores specifically for calcium to enter. The little bit of energy that calcium contains helps the neurotransmitters be released from the knob towards the membrane of the postsynaptic neuron. Calcium goes in, the neurotransmitter goes out. The neurotransmitter travels out of the knob into the presynaptic cleft, or the space between pre and post synapse. A neurotransmitter binds with the channel receptor on the membrane of the postsynaptic neuron, which allows for an ion, positive or negative, to travel through the membrane and change the potential of the neuron cell. So we see this guy come over here, and it opens the cell, and it allows positive or negative ions to come through. One of two results will happen from the synapse. Either the cell is excited and continues pushing forward action potential, or it does not meet the threshold for action potential and communication is limited. When thinking of the inhibitory synapse, think of how alcohol is known to make people have decreased reaction time. Since one cell has limited communication with the other cell, sometimes messages get paused or never get delivered. The reaction time for a drunk individual is due to the negative ions blocking communication. Excitatory response would be the opposite. There's enough positive energy from the ions to push forward action potential in the neighboring cell. Action potential, the sodium-potassium exchange through the membrane of a cell, reaches the axon terminal of a neuron cell. Voltage-gated pores open specifically for calcium ions to enter into the axon. The increase in positive energy releases neurotransmitter from the axon towards the synaptic gap between the dendrite of the postsynaptic neuron. The neurotransmitter then binds to a channel to allow for positive or negative ions to flow into the postsynaptic neuron. Positive ions lead to action potential being reached in the postsynaptic neuron and continues communication through the body. Negative ions lead to decrease in communication. That is how the body internally communicates messages, through electrical signals from action potential to chemical signals of the neurotransmitters and back again to action potential. Things like alcohol that would increase neurotransmitters to allow negative ions to flow into the postsynaptic cell decreases our reaction time due to the communication errors between neurons. Thank you very much for watching this informational video on the structure and function of a synapse. A special shout out to William Catterall, who has posted videos on YouTube via iBiology of the incredible work that he has done working on discovering the sodium channel protein. We'll see you again here next week for the next video. Thank you very much.